Hello, I'm Maruk Said, your microbiology guide. Let's explore in-depth micro techniques and uncover groundbreaking insight together on this educational journey into the fascinating realms of microbes.
Ribosome sequencing explained. Track protein synthesis at the molecular level. Table of contents. Introduction. Historical. Principle. Process. Advantages. Limitations. Applications. Introduction to ribosome sequencing. Ribosome sequencing, also known as ribosome profiling, is a technique that sequences ribosome-protected mRNA fragments to monitor the process of translation. Ribosome sequencing is also referred to as active mRNA translation sequencing or global translation initiation sequencing. It identifies all active ribosomes in a cell at a specific moment, providing insights into which proteins are being produced. This method offers detailed information about how ribosomes translate mRNAs into proteins, giving a clearer understanding of gene expression than traditional RNA sequencing. Traditional RNA sequencing only measures the quantity of mRNA present in a cell and does not reveal whether those mRNAs are being translated into proteins. The presence of mRNA in a cell does not necessarily indicate active protein synthesis. Ribo-Seq specifically targets ribosome-protected mRNAs that are actively undergoing translation. Understanding the process of translation is crucial for comprehending gene expression and cellular functions. During translation, ribosomes read mRNA sequences in codons, sets of 3 nucleotides, and assemble the corresponding amino acids into a protein chain. Ribosome sequencing is a powerful method for studying protein synthesis and gene regulation with greater precision compared to traditional approaches. This technique is valuable in many areas of biological research. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. Historical developments of ribosome sequencing. Ribosome sequencing was first introduced in 2009 by Nicholas Ingoglia and Jonathan Weissman. Their study demonstrated that sequencing ribosome-protected mRNA fragments could provide a detailed understanding of translation in living cells. Traditional methods used before ribosome sequencing lacked the precision needed to accurately determine ribosome positions, translation rates, and protein synthesis activity. Ribosome sequencing addressed these limitations by offering more accurate and high-resolution insights into active translation processes. Advancements in library preparation techniques and data analysis methods have further improved the accuracy and effectiveness of ribosome sequencing. Ribosome sequencing has been integrated with other datasets, such as transcriptomics and proteomics, to provide a more comprehensive view of gene regulation and protein synthesis. Traditional ribosome profiling methods cannot detect differences between individual cells, which is a limitation because ribosomes vary between cell types and can influence which mRNAs are translated. To study translation in individual cells, researchers developed single-cell ribosome profiling using a technique known as dual ligation. For profiling translation in small cell samples, a low-input ribosome profiling method has been developed using the template switch technique, enabling analysis with as few as a few hundred cells. Innovative methods like RiboMap have been introduced to further enhance single-cell ribosome sequencing by increasing mRNA capture efficiency. RiboMap was specifically designed to overcome the challenge of translation changes and cell stress caused by cell isolation, allowing researchers to study translation without needing to isolate individual cells. Principle of ribosome sequencing. Ribosome sequencing works on the principle that actively translating ribosomes protect specific regions of mRNA from degradation by ribonuclease enzymes. These protected mRNA regions are known as ribosome footprints, and their presence indicates areas of active translation. The number of ribosome footprints directly reflects the level of active protein synthesis occurring in the cell. By identifying ribosome footprints, ribosome sequencing enables the detection of actively translating ribosomes, which is essential for studying protein synthesis. Sequencing these ribosome-protected fragments allows precise mapping of ribosome positions on mRNAs and provides detailed insights into translation activity. The process begins by freezing ribosomes in place and treating cells with ribonucleases to digest unprotected mRNA regions, isolating only the ribosome-protected fragments. These isolated mRNA fragments are then converted into cDNA libraries that are compatible with next-generation sequencing platforms. The resulting sequencing data is analyzed to study translation dynamics at high resolution. Process of ribosome sequencing. Number 1. Cell lysis. Cells are first lysed to release mRNA molecules bound to ribosomes. This can be achieved using detergents, mechanical disruption, or enzymatic digestion. To preserve ribosome positions during lysis, translation is halted using chemicals like cycloheximide or by rapidly freezing the cells with liquid nitrogen. The resulting extract contains ribosomes, mRNA, proteins, and other cellular components. Number 2. Ribonuclease digestion. The lysate is treated with ribonuclease enzymes to digest all RNA not protected by ribosomes. This leaves behind only the ribosome-protected mRNA fragments. Enzymes like RNase I or micrococcal nuclease are commonly used to selectively degrade unprotected RNA, ensuring that only actively translated mRNA fragments remain for analysis. Number 3. RNA isolation. After digestion, the mRNA ribosome complexes are separated from the rest of the cellular material using centrifugation or chromatography. The protected mRNA fragments, known as ribosome footprints, are then size selected, typically around 28 to 30 nucleotides in length, using polyacrylamide gel electrophoresis for purification. Number 4. Ribosomal RNA depletion. Even after purification, the sample still contains abundant ribosomal RNA, which must be removed to focus on the ribosome-protected mRNA fragments. This is done using biotinylated oligonucleotides that bind ribosomal RNA, followed by removal with streptavidin-coated magnetic beads. Number 5. Library preparation. The isolated RNA fragments are converted into complementary DNA through reverse transcription. The resulting complementary DNA is then amplified by PCR to generate sufficient material for sequencing. Number 6. Sequencing. The prepared complementary DNA library is sequenced using next-generation sequencing technologies to produce short reads corresponding to ribosome footprints. Number 7. Data analysis. The sequencing data is processed to identify which mRNAs are actively being translated. This includes steps such as quality control, removal of adapter sequences, and ribosomal RNA contaminants, sequence alignment, and visualization of ribosome footprints to analyze gene regulation and translation dynamics. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. Advantages of ribosome sequencing. Ribosome sequencing provides a better understanding of gene regulation by identifying mRNAs that are actively undergoing translation. It can detect previously unknown proteins, small peptides, and alternative isoforms of known proteins. The method offers higher accuracy for studying protein synthesis by revealing the exact locations of ribosomes on mRNA, enabling precise analysis of active translation. It is especially useful for identifying low-level translation activity that might be missed by other methods. Ribosome sequencing does not require prior knowledge of RNA sequences, making it effective for discovering novel coding regions. Compared to traditional RNA sequencing, ribosome sequencing offers more detailed and focused information by targeting only mRNAs that are actively being translated. Limitations of ribosome sequencing. Ribosome sequencing generates large volumes of data, making data analysis complex and requiring advanced bioinformatics tools for accurate interpretation. It demands high-quality RNA and carefully prepared cell extracts to ensure reliable results. The method typically requires large amounts of RNA, making it less suitable for low input or limited sample sizes. Although sensitive, ribosome sequencing may struggle to detect low-abundance mRNAs with few ribosomes bound, potentially missing subtle translation events. The RNase enzymes used can have sequence preferences, which may introduce bias and affect the accuracy of translation analysis. The procedure involves multiple labor-intensive steps, such as RNA extraction, enzymatic digestion, and library preparation. Ribo-Seq requires specialized equipment and is more expensive than many other gene expression analysis methods. Applications of ribosome sequencing. Ribosome sequencing is used to identify mRNAs that are actively being translated by ribosomes, offering a detailed view of gene expression at the translational level. It allows observation of how newly synthesized proteins fold during translation. The method can be used to discover novel proteins and alternative isoforms of known proteins. Ribosome sequencing provides information about translation initiation sites, showing where ribosomes begin synthesizing proteins on mRNA. It detects locations on mRNA where ribosomes temporarily pause during translation, which can influence protein folding and gene regulation. It is used to monitor protein production and predict protein abundance in cells. Ribosome sequencing has applications in identifying translation-related changes in diseases by comparing translation activity between healthy and diseased tissues. Thank you so much for watching. Don't forget to subscribe to my channel, press the bell icon button. Thank you!